Hello class, today we will explore the oxygen dissociation curve, a crucial concept in understanding how oxygen is transported and released in the body. This topic ties directly into respiration, hemoglobin function, and physiological adaptations in different organisms. By the end of this lesson, you should be able to interpret shifts in the curve and explain their physiological significance. The oxygen dissociation curve is a graph that shows the relationship between the partial pressure of oxygen, PO2, and the percentage saturation of hemoglobin with oxygen. It has a sigmoid, S-shaped curve. Why is it sigmoid? Because hemoglobin exhibits cooperative binding. As one oxygen molecule binds, it becomes easier for the next to attach. This steepens the curve in the middle range. At low oxygen partial pressure, hemoglobin binds oxygen reluctantly. But once the first molecule binds, affinity increases sharply. At high partial pressure of oxygen, hemoglobin becomes saturated and the curve levels off. This graph is essential in showing how oxygen is loaded in the lungs and unloaded in tissues. Let's now consider the role of temperature. A rise in temperature, such as during fever or exercise, reduces hemoglobin's affinity for oxygen. This weakens the hemoglobin-oxygen bond and shifts the curve to the right, promoting oxygen release. Think of this as the body saying, "We're hot and active. Release more oxygen to the tissues." During exercise, muscles produce heat and carbon dioxide. These changes cause the curve to shift rightward, making oxygen more readily available to meet increased metabolic demands. This is a practical example of how the oxygen dissociation curve supports homeostasis under physical stress. Now we dive into the Bohr effect, a key physiological response. When carbon dioxide levels increase, hemoglobin's affinity for oxygen decreases. This facilitates oxygen unloading where it's needed most, in actively respiring tissues producing carbon dioxide. So in lungs, where the partial pressure of carbon dioxide is low, hemoglobin loads oxygen effectively. In tissues, where the partial pressure of carbon dioxide is high, it unloads oxygen efficiently. This dynamic is essential for gas exchange. Let's summarize. The curve shifts. Right when there's more carbon dioxide, higher temperature, or increased H+ acidity, this promotes oxygen release. Left in the absence of these conditions, this favors oxygen loading. These shifts help hemoglobin adapt to local tissue conditions. The fetus relies on maternal blood for oxygen. Fetal hemoglobin has a higher affinity for oxygen than adult hemoglobin. Why? Because it must extract oxygen from maternal blood across the placenta. This results in the fetal oxygen dissociation curve being shifted to the left, ensuring efficient uptake even at lower oxygen partial pressure levels. The fetus also benefits from increased maternal blood flow and a highly vascularized placenta. Now we compare lugworms, which live in oxygen-poor mud, with humans. Lugworm hemoglobin has a very high oxygen affinity, which allows it to bind oxygen even when levels are low. That's why its dissociation curve lies to the left of ours. Let's end with the metabolic consequences. Lugworms, due to their high affinity, hold on to oxygen longer and release it slowly, suited for low oxygen environments but not ideal for high energy activities. Humans, by contrast, have hemoglobin adapted for quick oxygen release, supporting active metabolism and vigorous activity. This highlights how hemoglobin has evolved to match an organism's ecological niche and physiological demands. In summary, the oxygen dissociation curve is not just a graph, it's a window into the dynamic relationship between hemoglobin and oxygen, finely tuned to meet the body's changing needs. Understanding its shifts helps explain everything from fetal survival to athlete endurance and even survival strategies in low oxygen environments. If you found this useful, subscribe, comment, and share. Follow us on our socials.